Okay, so in the previous video we were looking at what biodiversity is and how we can protect biodiversity. So let's have a look at how we can actually measure biodiversity using sampling techniques. Now we look at two groups of sampling techniques, if we're talking about plants or if we're talking about animals. Now the good thing with plants is they stay still. So they stay still, so it gives us more time to actually collect our data. The problem with animals is they move. They move from place to place, so we're not going to get a very accurate estimate of the population. Animals will move into an area, they'll move out of an area, they'll be very, very hard to count. Even some slow-moving animals like snails. So we tend to use a couple of techniques for investigating plant populations and a few techniques for monitoring animals. But what we want to do is you want to try to avoid bias at all costs. So we're not going to go to a certain area and just look there nowhere else. We need to find some random way of picking those areas, so we need to be random. And we're also in a large sample size so it becomes reliable, so we get reliable, accurate data. So one of the most common methods in terms of plants is using this thing called a quadrat. Now quadrat is basically a grid. You get different types. Some are just a square, some have small squares within. And the idea is you place the quadrat down on the ground. See a few different types here. And you randomly place them down. So you could come up with coordinates. So you could put some tape measures out of, like a letter L, almost like the axis of a graph. So you've got your Y and your X axis, say 20 meters by 20 meters. Use a random name generator or put numbers on a pieces of paper and draw them out randomly to select your coordinates, so say 5 and 6. So you move along the bottom 5 places and move up 6 and place your quadrat down. Count the species you're looking for. So you might be looking for daisies, or you might be looking for dandelions or buttercups. And then log that and then move on to your next sample, your next coordinate, and you keep going until you've collected sufficient data. Then using that data, what you can do is you get an average number of plants per quadrat. And if you know the overall area of the field, you can estimate the population size for that particular field. So if you know, for example, that you could fit 1,000 quadrats in that field, and there is an average of 4 daisies per quadrat, well, 1,000 times 4 would give you a population of 4,000. So that's your estimate. You can also use something called a transect. Now a transect is in a similar way, but this time we put a straight line, we use a long tape measure, we put it down, and we, again, randomly, we move along the tape measure. It might go every half metre, every 2 metres, every 5 metres, but we'll place a quadrat alongside and then we'll count the population. We're looking here effectively for a change of distribution of a species. So we might be looking on the inside of a woodland, going out to the centre of a field. We could be moving along the shore, on the coast, to see where species prefer to live. Do they prefer to live under the trees? Do they prefer to be out in the open? Do they prefer to be on the lower shore or the upper shore? We might use kick sampling in a river. So the idea is you have a net, and you kick the sediment at the bottom, and what happens is the water flow, the water current pushes the insects into the net, and then you can log them, so you have data on the number of organisms that are actually in that section of river, and then you can move along the river at certain distances. Now for bigger animals, we might use tagging processes. So you see there's a whale here that's been given a tag. You see there's a seal being attached here, a tag being attached to the seal here. And what this does is they have a unique number and a unique frequency, so we can track the movements of the animals, but we can also log the population size as well. So we learn an awful lot by tagging species. They don't harm them, they're very inconspicuous, they don't inhibit their movement, they don't increase the chances of being killed by a predator. It just helps us to monitor these larger animals. Whereas with plants, being stationary, we can use our quadrats or our line transect.